among our aging population, represents about 1% of all cancers, about 2% of cancer deaths. About 20,000 people are diagnosed with this disease each year. About 11,000 individuals are dying from multiple myeloma. And again, it's diagnosed through the presence of plasma cells in the bone marrow, and the progression of the disease starts with a pre-malignant lesion called MGUS, which is detected in a lot of individuals, and then it can progress on to a disease known as smoldering myeloma, and then progresses on to indolent and then symptomatic multiple myeloma, which is the really nasty disease that many of the people are suffering from. I'm going to skip through some of this stuff. And so in order to better understand the cause of myeloma, we need to molecularly characterize the tumor cells, the disease itself, to identify those markers that will help us better understand the progression of the disease. So I've been working very, very closely with investigators at Mayo Clinic Scottsdale, in particular Raphael Fonseca and Leif Bergsagel, and some technology companies, in particular Agilent Laboratories. We actually molecularly characterized a set of about 80 multiple myeloma tumors from 80 different patients, and we performed an extremely in-depth analysis of the genomes of these tumor cells in order to identify new markers to help us understand the disease and to help us understand why certain patients were responding to some of the chemotherapies and why certain patients were not. And this is just a figure of some of the data that's generated, which gives us a view of what the genome of a multiple myeloma cell looks like. And I don't have the normal picture, but in general, each one of these lines with the little lines through, these are the different human chromosomes. And so we can, with very, very high resolution, determine which regions of the chromosomes are actually abnormal as compared to normal cells. And this gives us insights on where in the genome we need to look for these genes that might actually be influencing the disease. And we identified this gene TRAF3, which we've now found that is extremely important in multiple myeloma. And we found that this gene is actually abnormal in 20% of multiple myeloma cases. And more importantly, it's part of a series, a very, very important molecular network in cells called the NF-kappa B pathway. And all of the black symbols are proteins that are generally normal in multiple myeloma tumors, but any one that's in red or green is actually abnormal. If you look at any person's, if you were to look at 100 multiple myeloma tumors, almost half individuals would have an abnormality in either one of these green or one of these red proteins. So this was a landmark study, and we went on actually to show that, more importantly, individuals who had abnormalities in these pathways were far more likely to respond to this new drug called Velcade or bortezomib, and this was the first study of its kind. And the cancer reviews in the same journal that this particular study was published in, they went on to say that although the exact molecular basis for differential sensitivity of individuals to this drug, bortezomib versus dexamethasone, is not clear, it appears that TRAF3 status, so the gene that we identified, TRAF3, which can be assessed fairly simply by conventional molecular techniques, should dictate treatment to Velcade. So basically what they're saying is if we can go in and look at these tumors and determine whether or not this particular protein or another gene in that pathway is altered, we can determine whether, with a very, very high degree of sensitivity, whether or not a patient is likely to respond to this drug or not. So we've also been working with individuals across the country in a very, very large study that's being funded by the Multiple Myeloma Research Foundation, or the MMRF. Many of you have heard of the MMRF and their consortium, the MMRC. We've started a collaborative research study that includes TGen and the Broad Institute of Harvard and MIT, and the Dana-Farber Cancer Institute of Harvard University and Mayo Clinic Scottsdale. And we formed a collaborative research team. And you can see that this is basically a dream team of genomic and clinical researchers to really get at the crux of multiple myeloma. And we're performing a very, very large study, and we'll be finished with the study at the end of May, where we will have data on, actually it'll be closer to hopefully 300 multiple myeloma tumors, where we will perform very, very deep molecular characterization, including this line where we're going to actually search every one of the 3 billion nucleotides, okay? in 200 individuals. So that work is going to be done at Harvard. And my laboratory is performing all of the microarray-based research. And then we're going to combine all of this data and perform a very comprehensive bioinformatics analysis to identify the compendium of genomic alterations in multiple myeloma. We're hoping that this information will provide us new insights to determining the difference between individuals who are newly diagnosed versus those patients' tumors who might have relapsed. And we're also hoping that it'll identify novel therapeutic targets for us to really be able to battle this disease better. And so, again, most of the microarray work is being done in my laboratory. It's being headed up by Angela Baker. This is just some of the data that she's been generating. Some of the data has already been analyzed at Harvard and the Broad Institute, where we've actually identified some of the well-known alterations, and more recently have actually started to identify novel alterations that have never been seen in multiple myeloma because of the large numbers of samples that we've actually been able to look at. And we were just recently funded to extend this study to perform a new level of genomic measurement called epigenetics, or looking for promoter hypermethylation in the multiple myeloma genome. And this work is actually being done in my lab by one of my very talented postdoctoral fellow who actually went to the Ft.defiance myeloma meeting, Badur Salhia, who actually is leading this work. She wrote the grant, got the grant funded through the MMRF, and will add to this data set. And also, I think Barbara mentioned, one of the interesting aspects of multiple myeloma that's been fairly overlooked is that multiple myeloma is one of the most significant health disparities, or the most significant cancer health disparities, where this disease has a twofold increase in incidence in men, among African American men, and as well as women. And with respect to mortality, rates, it also has a greater than twofold on average difference in mortality rate among African Americans as compared to European Americans. And I recently begun a collaboration with John Shaughnessy at the University of Arkansas, and we hope to actually analyze the genomes of about 75 African American multiple myeloma patients. And then we're going to have all of this data that we've generated through the MMRF study, and we'll be able to compare the genomes to see if there is an enrichment of particular alterations that we might see among tumors in African Americans that might help explain the differences in incidence and mortality that we see among this particular population. We know that socioeconomic factors, access to health may actually play a role in these differences, but we can't rule out actual biological factors until we actually do the study. So we're fairly confident that this work will be funded and we'll be able to actually contribute very significantly to this very important question. We're also, again, really going to analyze deeply the genomes of multiple myelomas using by DNA sequencing. And when I say sequencing, the human genome is made up, again, of 3 billion individual nucleotides. So it would be like taking a chain with 3 billion links in it. And any one of those links actually could result in a significant problem. So we're actually going to sequence and look at each one of those links in the 3 billion link chain to identify differences in multiple myeloma. And this work will be done at TGen in collaboration with David Craig, John Pearson, and Matt Huntelman. And we've already begun to generate some of that data, and again, I apologize for the darkness of these slides. But we've already begun to identify mutations that occur in these tumors. So I'm going to end right here. I know I only have 15 minutes. So again, TGen has built a world-class genomic and clinical research institute in about a 5-year period. And we hope to continue to be at the forefront of translational research, including in the area of multiple myeloma. So I'll stop there. Thank you.